Good.
So good afternoon. We have today Panagiotis Karanis from the University of Cologne and University of Nicosia in Cyprus. He will talk about the challenging about detecting and controlling Cryptosporidium. Sorry. So yeah, Panagiotis, thank you for giving us this interesting webinar today. Please, the screen is yours.
Yes, thank you very much, Zarena. Good afternoon to everyone. Thank you, Zarena, for inviting me to give this lecture about the topic about in vitro culture of Cryptosporidium. You will understand during the lecture why this is important, and it is the highest challenge now regarding Cryptosporidium research, and it is absolutely compatible to the cost action which is here presented to the ParAqua or aquatic system, because Cryptosporidium is waterborne. And the topics of my presentation, they are listed here. So the first point, it is about the future of infectious disease. I give the take-home message at the beginning of the lecture, not at the end. There are many other. Sorry, I increase now the size of the slides. Yeah, the second point, it will be few elements about my curriculum. It was a decision for life to work about this kind of disease, and this kind of disease, they are neglected disease with question mark, because they are coming again, emerging, and we call them re-emerging, are really neglected. It is correct to use the term neglected. And the point 4, this is about now the main material for Cryptosporidium and cryptosporidiosis, clinical aspects, the diarrhea and evolution of Cryptosporidium, and its recently clinical recognition, about 10 years that we have. I see that I have a mistake here, recognition in the word, about genetic polymorphism, about the detection and diagnosis and its evolution for monitoring on water and food, the methods which we are using since 30 years. And the last point will be about culture systems in vitro and in vivo. A few words about business scope. What is the commercialism from this basic research to shift to applied research and develop diagnostic tests. And I include also my international collaboration, because I want to give an education message to each one of us. So the future of the infectious disease, what is the future, not the past, because we are in the future now. So infectious disease, they belong to our life. They will be here forever, and they will never disappear. And the future of the infectious disease, this is the take-home message for people are interested and dedicated to this kind of research. There are many reasons why the infectious disease or mass outbreaks and infectious disease will affect the world populations, but in ever shorter intervals. It was a good situation in question mark, a positive situation, the education of people worldwide during the corona. So it is easier for me now to speak, and the people, they're speaking about such kind of disease, because the people, they understand. Many of them, they understand too much. They overestimate themselves. They know about PCI and they know the epidemiology. I don't need to explain what was difficult in the time before. So what is the reason that I say the future of infectious disease? That today's communities, they are influenced by water, food, air, biosolid contamination, wild animals, and environment and climate changes, demographic changes, and climate changes, they are going together nowadays. So these parameters, they are very critical. They are very important for pandemics. And they are natural phenomena related directly to ecological and demographic changes, what we have currently. And they are natural disasters. We experience this every time. It's just a question if you experience or not. It depends on which part of the world you are. We experience this even in locations in which they were protected or they were safe from this situation before. So because also of globalization and geopolitical instability and imbalance, we have the current situation. No need to explain further this situation. So it was a decision for life. Few words about my curriculum, where I started. I am original Greek, coming from Central Greece. Here you can see the small community, a village in which now only 10 people living there. And most of them, they are in the cemetery, in the margin, in the periphery of the village. In my time, we had maybe more than 100 inhabitants, and we call this the Greek Switzerland, because very beautiful, only mountains, and best clean air worldwide, second by UNESCO. And this is me here when I was little boy by the age of 13, together with my father, who play an important role for what I am today about infectious disease. Here I learned, I had the first lessons, the first messages about infectious disease, parasitic disease, particular parasitic disease, because we treated our animals by ourselves. No, it was not veterinary service. We needed to give the drugs, we need to give the vaccination, and also collected ticks at that time. Tick-borne diseases are very important nowadays. And I develop also another faith, another affinity for another science, for anthropology and anatomy. As children, we went to the cemetery. We scared a lot, not alone, so we were in group. And what you see here, the scalps and the bones, they were on the ground, not in the boxes as you see this today. And we were able, because the characteristics in the face, they are very conservative, not change the families, and we were able to guess who is who died. And as a children, we make a game like this. So I developed a big love for anatomy. What I am today, I want to let you know who is in your front, just to understand that you are speaking with a professor for parasitology in medicine, now 30 years having experiences in research, teaching on infectious disease, and I focus on, we call them waterborne, foodborne, vector-borne. These are the parasitic disease, but we focus mainly on this, on the detection from water, food, and vector-borne. This is modern. This is something what is needed. And I am also professor for anatomy in medicine, more than 20 years now, 23 years since 2000, teaching clinically oriented anatomy, unlimited dissections on cadavers, teaching neuroanatomy, embryology, and histology. So this is what we have in the basic clinical, in the basic and clinical part of medicines. I have accidentally and fortunately, I have dual career development and qualifications with long history in teaching in basic and clinical education. This is a message for everyone and also for the governments to put in experts in some positions which they need experts. And this is a problem today. When I reached the age of 60, I was wondering about myself, why it was so difficult, so actually painful, all the development, why it was so difficult. And I had the feeling that, like Hercules, who was the choice between the vice and the virtue, the vice and the virtue, kakia and arete. We learned this, everyone in the school. One was a life of pleasure, and the other one was hardship and honor. But it takes long time. And I think it was also with pleasure. Without pleasure, it would be not possible, because I developed a hobby for this kind of science I'm doing today. But it was very painful and very difficult, very difficult to convince the governments, the people. And the reason for this, it is because we call them neglected disease, which are today emerging and re-emerging. But if you are working with neglected disease, you will be always neglected. I feel always neglected in my life, but suddenly the people, they found this very interesting. And this is a dilemma. I think as educator, the problem is the definition of the pathogens. They are not neglected. This is a discrimination inside of the science, and this, of course, necessary. I never had the time in the past to write an article to the Nature in order to change the situation, to neglect the neglected, in order to come to the point. And there are a list of neglected pathogens. I'm not going through the list now, but in this neglected pathogens, through my teaching in medical parasitology and tropical disease in different universities worldwide, I found that the parasites, they are overlooked and unloved. So the field of parasitology is always parasitized by people. They are looking for opportunity, research opportunity, and this is the world of the parasites, which is a fascination. And I wonder it when it comes to the Nobel Prize 2015, how to call these parasites neglected parasites, which affect millions of people. And this is what I said before. It is a discrimination inside of the scientific community, because the people, millions of people are affected. So we cannot call them neglected parasites because they are in the so-called third world countries. And in the year 2015, 3 people shared the Nobel Prize here for the treatment of parasitic disease, and there's still a lot of potential, what is very important for us. I think that the countries, they should establish negotiation groups to explore pandemic and outbreak prevention, because it's always ongoing situation, and the countries need to be prepared to respond in the time when the outbreaks, they are coming. So when you remember, when COVID started, the first point, it was to know if the people, they are infected or not. And this was the necessary in the first week by COVID. Some experts, particularly from Germany, they develop immediately the test to apply if the people are infected or not. And we have the same situation here. Therefore, monitoring, diagnosis, and surveillance. And this is what I represent from the medical point of view, the development here of the diagnostic methods to support the healthcare system, which they need to expand to detect and control the infectious disease. Many of them, they are undetected, but it's also development and exploration of an unlimited opportunities for jobs and for research, which I call now the golden era of infectious disease restarted. I continue now about Cryptosporidium, the main topic. I take this pathogen now as an example. There are other pathogens, but I focus today on Cryptosporidium. Why? You can find Cryptosporidium everywhere. Based on the One Health concept, we need to have healthy environment, we need to have healthy animals, we need to have healthy humans. But this is not something new. The One Health concept, it is not new. It exists since Hippocrates, and it has been reintroduced in the 40s by the Canadian doctor or US doctor, veterinary doctor, and in the as a public health model. And now many colleagues, they discovered this new. It sounds very good, but it's not new. So water bodies, food and air, they are contaminated, and they are exchanges and connections between the environment, between this material, food and water in the air, and the humans and animals, they are infected. If we want to be healthy, if we want to have healthy animals, we need to have healthy environment. And the contamination and pollution, including the air, Cryptosporidium is not only waterborne, foodborne, but it's also airborne. I will explain a little bit. And this model from the colleagues from China, where I contributed a lot in the last years, that this kind of things they develop, and unlimited papers coming now from China, because the government spends so much money for this kind of the research. Here, the colleagues from China, they show us the health risks from Cryptosporidium and Giardia. I put here also Giardia because we learned a lot of Giardia before. When we shifted to Cryptosporidium, what I say for Cryptosporidium may be also valid for it is valid also for Giardia, but we focus today, for simple reasons of the time, only on Cryptosporidium. And you see here that there is contamination because of the fertilizers, because of the effluents, everything is going to the water, to the surface water. And if you think that the River Rhine in Germany, for example, is going more than 1,000 kilometers, and right and left from the River Rhine, many cities, they take the water from the River Rhine, we can prepare, we can treat the water, but it's contaminated. And what we can do, we can reduce the pathogens. Some of them we can kill. We cannot kill Cryptosporidium. And here you have also the situation with the agriculture people who are the, so we have with the consumers, we have the farmers, and this is another topic here for the agriculture practices, because Cryptosporidium and Giardia, they are distributed through the agriculture practices, and the people here who reported this situation, they wanted to alert the community, the medical community, for the cancer patients, and they are susceptible subpopulations, and there is a risk mitigation scenario analysis on the pretreatment of surface water supplies and the feces which are distributed in the environment, and the production of the vegetables which are contaminated. Why Cryptosporidium research? This question now, it is easy to answer. I don't need to convince anymore to the Ministry of Health. I don't need to convince anymore the Ministry of Environment. Years ago, it was the situation that we went to Ministry of Health. He said, no, this is not my responsibility, the Minister of Environment. And fortunately, the Ministry of Environment spent us money for the research when we started 30 years ago. And 2012, it was a paper which has been supported by Bill Gates Foundation, and Cryptosporidium was one of the 4 main pathogens responsible for severe diarrhea and mortality of children, particularly under 5 and 2 years, and was second most important diarrheal pathogen after rotavirus. So the number of the species which have been investigated, it was, I believe, 40, and it was very accurate project published in Lancet, and since then, 2012, Cryptosporidium reached its clinical reputation, and we can show this data thanks to the Bill Gates Foundation, who spent this money for this research for the children affected with diarrheal disease, responsible for 10% of the overall child mortality. And this takes a lot of interest and priority worldwide now, the diarrheal disease. Cryptosporidium was known as an opportunistic pathogen before. Only the HIV patients and people who manage the water supplies, they say, okay, we can cook the water, we can heat the water and give to the HIV patients or to the organ transplantation, to the people they get here organ transplantation. But this is not the only case, because Cryptosporidium doesn't affect only children, weak children, and also the immunosuppressed, but also the immunocompetent patients are affected, and there are a large number of such kind of patients, and there is a large number of waterborne and foodborne outbreaks. 60% of the waterborne outbreaks reported worldwide, they are going back to Cryptosporidium. About 40% are going to Giardia. When I focus only on the protozoan parasites, I excluded now viruses and bacteria, and they are about 1,000 outbreaks which are reported. And most of them, they are not reported. There are more millions of disability-adjusted life years. And now this is the point, there is no vaccine, there is no therapy, there is no drug and vaccine development because of the poor tractability of the parasite. So the systems, all of the cultivation systems, they failed to succeed here. And we can use some animal models. Maybe it's not correct that I say lack of animal models, because we have, but they are cost effective and time consuming. This is the problem. And in the meantime, also we have molecular tools which we can apply, but all of them, they have many disadvantages. And this research I transferred worldwide, invited by the by the Japanese government for several years, was visiting and full professor teaching staff in northern of Japan for the National Research Center. And I spent also years here in the Tibetan Plateau, where I established a biomedical center for infectious disease, coming back from China, going to always to the direction to Greece. I make a stop in Qatar for 2 years, and it was an invitation in the meantime from Cyprus. So I speaking now to you from Cyprus, and I proceed now with the Cryptosporidium pathogenesis, what happens in the intestine. When you see here the intestinal cells, you can see here the epithelial intestinal cells with the this is a crypt, and we see here the epithelial cells here, this is the crypt, and these are the epithelial cells, and you see the nuclei, which are basal, the nuclei, and you see the microvilli. You see the black dots here everywhere. These are stages from the very complicated life cycle of Cryptosporidium. We cannot say exactly which kind of stages, because all of them, they are same in the size. We have, for people they know about coccidia, they have trophozoites, sporozoites, trophozoites, merozoites, and also the sexual stages, and all of them, they are looking the same here. I cannot say, but this is a person, HIV person, who was affected, and this is a biopsy, and you recognize much better now what happens in the microvilli. These are the microvilli of the epithelial cells. Here, this is the normal light microscope, and this is electron microscopy, where you recognize one stage, which we call trophozoite. It is one cell. It is about 4 micrometer inside or less, and it has inside the nuclei and the cytoplasm. This is another stage. We call them merons. They have you see here, they have more than 6 or 8 stages, which are infectious, and there is a reproduction, autocycling here, recycling of the pathogen in the intestine, destroy here completely the microvillus and the Cryptosporidium. I have to say that it's not intracellular. It is an epicellular parasite, but it is extracytoplasmatic. Intracellular, extracytoplasmatic. The correct term, it is epicellular. You see here in the electron microscope, this is the picture in the electron microscope on your right, and this is the normal, also electron microscopic picture, but you see the trophozoite. Around here of the trophozoites, which is now established, in fact, here the intestinal cells, it is a parasitophorus vacuole, which is established after the infection, and it protects the parasite. So the parasite is not affected from this infection, but the integrity now of the tissue and the cells which are next to each other, these parallel cells, which are here, you can see the mitochondria, the integrity of the cells, it's lost, and the nexus between the cells is lost, which rely to the terrible diarrhea. That's why we call this the malabsorption syndrome, and it might be that the infection can be asymptomatic and relate to the chronic infection. But the most common clinical manifestations is profuse watery diarrhea, terrible diarrhea for people who are affected. Immunocompromised people, they give only water, they excrete only water and some bacteria and these parasites, and the diarrhea containing mucus and rarely blood and leukocytes. Other symptoms, they include the nausea, vomiting, cramp-like abdominal pain, and mild fever. And there's a group in France, in Lille, who they focus on this here research, and they really confirmed that Cryptosporidium appears to be associated with a human digestive cancer, and Cryptosporidium is the etiological agent, not the adenoma, which is present in cancer. Cryptosporidium, it has been confirmed in animal models, And it has been also confirmed that some people, they have cancer, they have high rate of Cryptosporidium infections. They investigated biopsies and they compare with people not have cancer, they don't have cancer, and they don't have Cryptosporidium infections. This is another point which is promoted in the last years, and we believe that there are some strains of Cryptosporidium which are more virulent. So they are carcinogenic strains of Cryptosporidium parvum, which are virulent. And this now again, I use the situation to say that COVID taught the people a lot of the things about the strains, isolates, and different genotypes and subtypes. And this is the situation also for Cryptosporidium. And here Cryptosporidium is a significant, an essential problem complication in recipients of organ transplants. And this is an underreported prevalence. Maybe by the next month, you can see our details, the details in a review, which is now under review, and it will come that we have this information underreported prevalence, and most people affected, they are people who get the kidney. Few words now about the immunology, what happens in the intestine from the immune response of the host. You can see here these stages of Cryptosporidium, which are now the sporozoites, the inside of one oocyst, we call the oocysts, which are transmitted fecal orally, and they excrete the sporozoites. The sporozoites develop in trophozoites, and there is a sensing. The first response is sensing here, which promote here the immune response in the lamina propria. And there is, of course, we need the innate immunity and later the adaptive immunity in order to be protected, but the immunity is a problem here in the Cryptosporidium. I will not go to the details. It's possible to look on this and also to ask later, but here the immune response, for the adaptive immune response from the host, it restricts the parasite also to entry the cells. As I said to you, that the nexus there here in the gap junctions between the cells, it is destroyed, and we have here disintegration of the tissue. Another important information is here that this parasitophorus vacuole protect the parasite from the immune response, and also there is an anti-apoptosis substances which are excreted here, like survivin from survival and osteoprotegerine, which first they protect from the apoptosis, the anti-apoptotic factors, but later they cannot work, and the gametes, the sexual stages, they are then they go to fertilization, or the cells, they are destroyed and they are released. No more details about their immune response. Question is how many Cryptosporidium species? How many Cryptosporidium species? When I started to learn about this 30 years ago, we know only about one species, Cryptosporidium parvum. It was simple Cryptosporidium parvum. Today, we have 47 recognized species and more than 120 confirmed genotypes. So the term isolates, genotypes, subtypes, and something like this become more familiar to everyone because of COVID situation. And they are about 20 species. This is now interesting regarding for humans, which are pathogenic. As I said before, 30 years before, we talk only about Cryptosporidium parvum. Today, everyone is talking about Cryptosporidium, and we have Cryptosporidium hominis, Cryptosporidium parvum. These are the most significant, but they are in the meantime 20 species of Cryptosporidium which have been identified in humans. And here, this is an evolution of Cryptosporidium species. Few words about this, few aspects. You see there's subclades, clades of the related to the comparative genomic survey of Cryptosporidium species worldwide, and they are cryptic anthroponotic. Of course, the people from the bioinformatics, they confuse now the community. They produce very excellent data, and they show that the Cryptosporidium branches, they have superclades of species and subtypes, which they undergo genetic exchange, potentially facilitating host association. So we see here, Cryptosporidium only humans. This is not correct. The Cryptosporidium hominis, it has been found also in animals, because they, some years ago, 20 years ago, the researchers, they said only in humans, but this is also in animals, probably due to its evolution. We have also humans and rabbits. Here, the species which found in travelers from rabbits, and we have Cryptosporidium parvum, which we call Cryptosporidium parvum anthroponosum, and it is categorized here with the number 2 in Latin. So the number 2 is for Cryptosporidium parvum anthroponosum. It causes anthroponosis in humans, and we have Cryptosporidium parvum, which is found in several humans and the livestock here. Different subtypes, and this is the evolution of Cryptosporidium species. And another information regarding the distribution of Cryptosporidiosis worldwide. These are the images B and C, and when you're looking in the left here on the B, you see the different colors. We have the blue and we have the orange. The blue one is about the distribution of Cryptosporidiosis because of Cryptosporidium hominis, and the orange one, it is Cryptosporidiosis due to the Cryptosporidium parvum. In the image on the right, this is here, the green one, it shows the Cryptosporidiosis due to the Cryptosporidium parvum, which is exclusive zoonotic, and the purple one, it is because of Cryptosporidium parvum anthroponosum. Yeah, so this distribution, of course, it depends just simple because of the published papers. Some countries, they don't publish, and many countries they don't publish, but this is a useful information to see about the distribution, and this will change. It is always ongoing and on change procedure, because new papers, they will come, new information, they will come. This is limited maybe to 80 papers or 100 based on the availability of the information, just to have an idea. And please remember that we have some pathogenic species which we didn't know before. We put them in pathogenic, and 20 or 30 years ago, we know only about Cryptosporidium parvum, but now we have 20 species which are pathogenic for humans and, of course, for animals. And this is the dominant species, isolate and genetic line, which is found now in the USA. The researchers, they say that this become the dominant Cryptosporidium hominis subtype in the USA, originate from subtypes from East Africa and from Europe, and it gives this dominant subtype. And you see here that we put in this isolates, we put the number 1 in Latin. This is the characterization or the description of isolates Cryptosporidium, which they belong to Cryptosporidium hominis. Cryptosporidium parvum with 2, Cryptosporidium hominis with 1. You remember, with COVID, we needed to use almost all letters of the Greek alphabet, some remaining, and here we have another categorization. So what happens in the Cryptosporidiosis in the stool of the HIV patient? You see here, this is the excretion, what I said before. They have watery diarrhea many times per day, and you see little material here, maybe mucus or fecal. You see some bacteria, and you see this round here images, which they have a very tiny cyst wall. It's very strong, that's why difficult to kill. And inside, in the cytoplasm, we have a mass. This mass we cannot recognize, but here maybe you can recognize little bit banana-shaped sporozoites, which are possible to recognize much better by specific methods induced here, the so-called excistation. You see the cyst wall, the oocyst wall. They are 2 oocysts. They are 5 micrometer in size. Inside, you see here some rest body. They are proteins, which they use the parasites to survive outside of the host. And you see the sporozoites, which they are 4. Inside of the oocyst, we have 4. It means we have 1 oocyst we cannot recognize, and it means we have 4 sporozoites, which are 4 infectious stages. If you have 10, you have 40. If you have 100, you have 400. And that's why the low infectious doses because of Cryptosporidium, and the infectious doses because of Cryptosporidium, it is about 1 to 10, or let's say, let's be a little bit more pragmatic, 10 to 100. This has been confirmed by volunteers, by animals, by humans. And these animals, they are infected. They are reservoirs for Cryptosporidium. They excrete billions. So 2 grams of this fecal material here from the cows can contain billions of Cryptosporidium oocysts. And this is not the correct farming, the rules for the correct farming. Some of the animals, they have also bloody diarrhea, and they can infect each other here. And you can imagine what you have 3,000, 5,000 of this kind of the animals living in a very small place and space. They can infect each other, and they are reservoirs for different species of Cryptosporidium. You can believe me here. This is fecal material from the cow. Directly on the microscope, you can recognize nothing. I put here a red arrow because I know that here there's some Cryptosporidium oocysts 5 micrometer in size, and I can tell you there are billions undercovered here in this fecal material, which is impossible to recognize, to see in the microscope, and we need to visualize them. And this is here what you see, 5 micrometer in size. This is the specific staining, Zil-Nielsen modified Kinyu staining, which we adapted from tuberculosis bacteria. And you can recognize also the size, but also it needs special expertise to recognize these parasites and to differentiate from other, for example, fungi, something like this. So the most time we spent in order to visualize this, because it's very difficult to diagnosis. And this is one weak point worldwide. You see here, it is a little bit higher magnification. The oocyst, which is fixed here, is fixed, and this the red mass, it is inside of the oocyst wall. And in some species, some hosts, it is possible to recognize. This is the best picture I ever had, the best image I ever had years ago from the snakes. And most probably this is some kind of maturity of the oocyst. Everything what is looking red here, these are oocysts, which are about 5 to 6 micrometer in the size. And I can recognize very clear here, there's banana-shaped sporozoites, and I said they are 4 inside of the oocyst. The transmission of the Cryptosporidium, it is fecal orally transmission. It is waterborne transmission. It is recognized, accepted, and reputed. It is foodborne transmission. Many years we tried to convince that it's a transmission way. Vegetables, ready-to-use vegetables, could be contaminated. And if you think in countries where they use also fecal and sewage material to irrigate here the vegetables, it is very simple to imagine. But this is also, it has also airborne transmission. About these chapters here, about these facts here, there are reviews which we prepared, and some reviews, they take more than 4 years sometimes in order to finish them. And there is a data, recent data, and updated everything you can read in the details. And I said the 3 modi of transmission: fecal oral transmission, waterborne transmission, foodborne transmission. And we are going now here because we found also people in their sputum. We found Cryptosporidium. Many people, they believe also that they go to the lungs. It is an airborne transmission. For example, here, the methodologies, they are very inhomogeneous worldwide because there is no agreement what each laboratory is doing. And this is one point alerted in the beginning, but these are here the details about this review. For example, it took more than 3 years to complete about the comparison of the current methods from the literature, and this was a meta-analysis, and this large disagreement in the methods. Coming now to alert the water community about the waterborne transmission of the Cryptosporidium and what is to do and what is the relation with the water treatment. We have large number of outbreaks. So the countries here mostly developed, they have high economic and sanitation standards like Europe, but Europe is not equal Europe. We have countries which they don't have high standards. We need to keep this in mind. But we say Europe, North America, Australia, they have well-developed economic and sanitation standards. Most of the outbreaks, they have been reported from those countries. So Sweden and Norway, they have outbreaks. We didn't know this before, but they have been discovered because it is well-developed public health regulation, high standards of public water supply and wastewater management. But the Cryptosporidium oocysts, they penetrate the filters. We know that flocculation and filtration, they can remove a lot of these parasites, but they only can reduce the initial number. And the oocysts, they are present in any kind of surface water worldwide. We have enough epidemiological evidence and confirmation about this in any country of the world, and also molecular epidemiology. And according to this, monitoring is of significant importance for the public health risk management. So the efforts we have done in the last 30, 40 years about the understanding of the occurrence, distribution, the sources, the fate of this organism in the environment, they are enough that we can say we have enough epidemiology now. We know these pathogens, they are distributed everywhere because wild animals, domestic animals, and any other animals, they are infected. And regarding the monitoring, how to monitor, how to find, how to detect this parasites, this Cryptosporidium in the water, it is the information which we have from the most accurate and reproducible standardized procedure. But they are not standard. I want to say also they are not standardized because different laboratories using it in different way. Monitoring, we recommend the Cryptosporidium should conform to widely accepted protocol, which is one USEPA 1622-23. It's been updated for many years, and the ISO 15553. But what is the mistake here? The mistake is widely accepted. I would correct in widely promoted. This is widely promoted, but not always accepted. Just countries or experts, they were not included in development of this kind of the methods and to the protocols. Therefore, this kind of the protocols, they have a lot of deficiencies, but this is what we have, even so many years after the first monitoring starting. So the interpretation of the data produced by other procedures, which they exist, and they are simpler methods. I can find Cryptosporidium, for example, in 1 liter of water, not needed to filter 50 liters or 100 liters of water. But these methodologies, they need to make their evolution to be combined with other more modern technologies and simplified molecular methods to combine and to complement. The Cryptosporidium data mostly it should be consistent, and only zeros, they tell very little about the situation. This is a misinterpretation, and this it brings to the dangerous misinterpretation that the organisms are absent. This is not true because this is under the detection limits. It depends on the methodology, and there is a variability. So here some mistake happens here, but doesn't matter. I will continue. So the raw numbers without consistent measurement of the recovery cannot be compared because of the significant variation in recovery efficiency. We say recovery efficiency. We don't have 100% recovery. So because of the different quality of the parasites in the distribution, because of the different quality of the waters, and because of the different numbers. So wastewaters are contaminated, not needed to investigate 50 liters of wastewater. 1 liter of wastewater could be successful if you combined efficiently with other methods, but the recovery efficiency of the organisms, it is a very important issue, and it produces also distrust in the governments during the years. What is about the ability to manage the watersheds or the treatment systems? We need to know all of the Cryptosporidium types, and not only the viable fraction. Informations of those futures of the organisms, they are present in any sampling location or time period, be useful for other purposes. So the pathogenic, we cannot discriminate between pathogenic and non-pathogenic. We need to detect all of them, and we need also to know the futures and the characteristic in the sources of the catchment areas to monitor the organisms. And everything what we have done until now, it was important to improve the water quality, the final water quality, but also the protection of the water supplies and watersheds. So the management of the water treatment facility it needs the knowledge of the total concentration in the raw water. This is an important issue, to know how much contamination we have in the raw water. What is the variation? And the variation, it depends on the recovery efficiency, it depends on the concentration during the annual cycle, precipitation, and many other factors which probably most of you they know. Then the last point, not in the value, but which I mentioned here, it is about the cost. The costs for monitoring Cryptosporidium, they must be efficient. And this is possible today if we combine the knowledge which we have and adjust the different methodologies in the most efficient methodology, simple and not very expensive. So the evolution of monitor for Giardia and Cryptosporidium, who wants to read all the parameters, all the details, it has been summarized in this in the year 2017, and you can find all of this information here in this review. As I told you before, it takes more than 3 or 4 years to finalize such kind of reviews. I come now to the point in which I want to connect the water treatment and the parasites in the lab, and also all the points which I mentioned before in relation to the One Health concept. So they are animal models. We cannot say we have animal models, but the problem is we can infect animals, small animals, to reproduce, and many colleagues from my time when I was PhD student, they have companies, now established companies in USA or in Australia, because it was a political situation every time, and the people, they lost their jobs, they needed to go to other jobs, and they established animal models to reproduce the parasite, and they are needed to, it is needed to disseminate the parasites for the animal models for the experimental purposes. We use also cell lines or organoids in the in vitro in order to reproduce the cells. And we use also in vitro axenic system. The problem is for the for the first one, it is here for the animal models time consuming, and also I do not propose, if it is possible without animal models, time consuming and very costly. Here regarding the in vitro cell lines, and this is a problem in the literature, they are susceptible to genetic variation. It's looking nice when you see the paper, but what it is contained inside of the papers, they cannot maintain all the phases of the Cryptosporidium parvum life cycle, and most cannot maintain and propagate the infection for extended periods of the time. This is the problem for the Cryptosporidium cultivation using cell lines. It is not reliable. The cell lines do not recapitulate the native intestinal epithelial tissue of the C. parvum host. This is another problem, and this encourages the search for increasingly accurate models of the study. and the organoids. This is a new technology, just started some years ago, and it is applied now for the infectious disease studies. And this is under development. But the benefits of the culture model are enticing and allow questions that were not possible to investigate with previous models before, still not established. What these are organoids, so we can create organoids. We can cultivate small intestine, small brain, small lungs, and we can try to. It has been here tried and published in the Nature paper, Nature Microbiology. The colleagues here from Netherlands. The important thing in this paper was that the people were able to bring the organoids in relation to Cryptosporidium development in vitro. This was the most important information, not another big achievement in the paper. But they use here also my picture, which was the cover in the International Journal of Parasitology. I told you before, here the explanation about the sporozoites, and you see also here the tissue here from the lungs, from the bronchi, where you see Cryptosporidium inside, and the airborne transmission of Cryptosporidium is most likely. So this is the situation using here the Cryptosporidium. We do in vitro excitation. We have the sporozoite, which they are transferred to merozoites, and we differentiate between small intestinal and small lung organoids. So this is a development, but didn't establish all of these papers, which I didn't show before, about cell lines and also about the organoids. They didn't repeat by themselves. We cannot use them to produce for a long time the pathogens. And what I promote, this is my system here, the axenic in vitro cultivation, because this is maybe the best way if it is working. It is under development. It is working in part. It needs further development. You can see here all of the stages of Cryptosporidium in vitro. So can you imagine that we have 10 oocysts, 1 oocyst, and we can develop, we can produce in vitro here millions of this kind of cells. This is the point, what we want to do. And this was not sustainable with the other models before. And this system is here, needs further development. All of the stages, I will not go to the details. You see here, there are cell divisions. There are all of these stages here. They reproduce, they make recycling inside of the intestine, in the intestine. And this is possible in the in vitro axenic cultivation. And this is the connection now with the detection in the environment, that you have very small amount of the oocysts and you can cultivate in vitro. This is the big advantage in the future. And I show you another image or other images here where you can follow here the reproduction of the trophozoites, which are the 3 to 2 micrometer in the size, and they can develop further. Here they divide, they are aggregates of trophozoites, and they like specific food, specific substances, specific treatment, also specific time of dedication to these parasites. You need to sleep with your parasite if you want to be successful. Don't leave your parasite in the night alone. Please follow in the culture, and maybe you have more success than you believe. So here we have also the merons, and this is the connection now. Again, I repeat, from small amounts of the parasites in the environmental samples to produce. Why? Yeah, I summarize. It was, I will explain why. I summarize, it was an invitation from the editor here in Parasitology, because there was a confusion in the medical community, in the scientific community, about the in vitro culture of Cryptosporidium using cells, organoids, and axenic. And all of the details, they are summarized in 2018. And we have some progress here. Yeah, now I'm going to explain what is the benefit, and I will come to the end very soon. what is the benefit with the in vitro cultivation? It is a huge amount, a huge potential for development, for business, and also investment. The diagnostic value and for any purpose, the countries, they can produce also these tests. Here we are in the laboratory, so we need to cultivate our parasites. We can handle with the parasites in the in vitro culture, and their applications in the human medicine. They are applications in the veterinary medicine, in water and agriculture industries. So today we bring in association with the water detection and the water treatment, and next time maybe with agriculture industries. This is possible for development of the quick tests, positive controls, the arsenic culture, the propagation, and viability tests. Millions and billions of euros worldwide, they are now invested for the detection, for the disinfection, for therapeutics and vaccines, for prophylactic and therapeutic purposes, for the development of drugs and evaluating here disinfections methods. This is now the future, but we need the in vitro mass cultivation system, which I used to teach in the all of these issues, what you saw here before, worldwide in different countries, and mainly in Germany, but also Japan, China, Greece, and Cyprus now. And the most of the data we have arise from the epidemiological studies. We have epidemiological studies. I don't put priority. If you have the possibility, you can put clinical studies first. But in our case, the epidemiology was the first part. So the people ask, do we have these parasites in our watershed areas? Do we have them? Yes, we have them, and we know now that almost all the animals, they are infected with Cryptosporidium. Other here knowledge arise from the in vitro cultivation system, which is very convenient in a small tube to have billions of the parasites in the lab, from the animal models, which I don't propose, but we need to many times to use. And we have also the possibility for new tools for genetic manipulation, but we need to be carefully what data is produced for any research group, because most of the research groups, they are dependent on the funding, and they produce papers which sometimes you read them, they are written very well, but when you're going to produce, it is very difficult. And of course, we're going to the clinical studies. We are much far from this situation. So I established a very large international working net worldwide, which is effective, is working every day from 4 in the morning until 11 in the night. There are people, young people, they like to collaborate. And this is also the reason why we come together here. And I hope that we can have also fruitful cooperations, develop new. Some of them, they have, of the people, young PhD students, undergraduate, postgraduate students, they came to the lab and they are professors in their own countries now, or we still continue now. We can send in a few seconds the images, exchange the data, and as we are doing now here. So this is interdisciplinary research, worldwide dissemination of the results, and one positive aspect is also the recruitment of collaborators. When you work 10 years with people who know, you can use also recruit for your research because they are experienced, and at least the intercultural exchange, and I have the good luck to get good experiences on this. Yeah, there were some images which I cited the sources, some informations they under the slides when you use in the PPT form, maybe not visible in the PDF form. And this is potato from the Greek supermarket, which I discovered. You see now here that I am anatomist, real anatomist, not only parasitologist. And these publications, and they arise from different internet sites, and they are property of their respective owners. But the most material, it was from myself. Thank you so much for listening to me.
Thank you. Thanks, Panagiotis. Yeah, it was very complete and interesting. Certainly a new point of view, a new parasite for us to discover. Yeah. Other question for Panagiotis? Maybe I can just start with maybe a bit trivial question, but well, you know, we work mainly with zoosporic parasite that infect algae and photosynthetic organism. So how do you see a bit the link of your work, which is mainly on human parasite, to ParAqua?
I think that here the research cohort or the research direction is going to develop some technologies, detection methods which are adaptable. And I think the algae for us, it will be a problem because we need to clean the samples, and the eutrophication, the algae, it came because of the different excretion from the agriculture, which bring also the Cryptosporidia. I don't know, maybe someone who is expert in the audience and he has a good suggestion how to remove. Maybe it is a possibility by the help, the support of the algae, also to remove pathogens. We cannot work selectively. We cannot say that we remove now from the water only one parasite. We need to remove all of them. So what is possible by flocculation and filtration, and maybe in the same projects, another point is when they are same projects to investigate water supplies, that we can include samples for Cryptosporidium. This is a link between the different participants. I can understand if this material is I don't know how to say, maybe some people are working only on the algae, and this question, it was a problem always that the people, they don't understand about the parasites because they didn't learn, and it is difficult also to make some questions. I see the cooperative work between the participants. When you are going to make some projects, we can also develop the techniques in parallel, or we can use samples from the same areas, from the same samples, in order to develop what I said in the last points about the cultivation of the, maybe also algae, they can support the cultivation of Cryptosporidium, co-cultivation, something like this.
Yeah, Hans-Peter, I let you talk. Just want to ask a precision.
Yeah,
so
I have a related question. So, in principle, zooplankton can filter out the Cryptosporidia, especially the spores, quite efficiently. So, what do you think? How does food web structure impact the presence or the survival or the disappearance of these parasites?
Maybe I will ask back, because I don't know if I understood correctly. You say the food material, how this affect the, if I understand the correction, the question correct.
No, I'm asking how the food web structure. So how much zooplankton, phytoplankton, and other trophic levels you have in aquatic systems may impact the survival or the disappearance, the clearance of the parasites.
Yeah, this question, I don't have any information about this. How we know that biofilms, they support the survival of pathogen parasites, and Cryptosporidium can survive outside in the environment for 2 years, a long time. If you take them, you go to the lab and infect the animals, you can see that they are infective, even you put them in the refrigerator. So they have a good survival. But what is exactly the coexistence and the exchange between them, I don't know, because really maybe no data about this. I'm not sure.
Yeah, can they be adsorbed to particle surfaces and then somehow be protected from grazers which would feed on the parasites?
Theoretically, yes. I would like spontaneously, yes, but we need to investigate to confirm this in the practical experiments. Yeah.
Okay.
Yeah.
Thank you very much.
Maybe I will complement a little bit. What we found, when you have the drinking water treatment, you have the surface water treatment. So the phosphorus eliminating plants in Germany, they have been developed in order to remove the algae, because in the 70s, it was a lot of contamination. It was in drinking water, a lot of algae, which contaminated the drinking water. At that time, the people didn't know about crypto. But later, what happens, despite the algae, they have been removed from the drinking water. Cryptosporidium, we can find Cryptosporidium much easier in the samples, and they can survive very well because we feed it the animals and they were infectious. The problem is that chlorine doesn't kill Cryptosporidium in the concentrations which is used for the water treatment. This is another aspect which may be of help about this question you addressed.
Yes, thank you very much.
Yeah, I just wanted to come back shortly about the method that you mentioned, and if you have some idea or some method that you apply regularly that can also be helpful in detecting different parasites, or how the method you use can help us in working with the zoosporic parasite.
You mean the detection techniques?
Yeah.
Yes. So the first one is this here, what I showed here in the microscope. And when you have high contamination, you can find the parasites. But the experience was more than 30 years now. There are a lot of fake results, wrong results. Really, you need expertise. The problem with Cryptosporidium is exactly the diagnosis. We had also outbreaks that people, they thought they were outbreaks, and in reality, they were not Cryptosporidium, they were algae. And this was a big issue once happened in Australia. And the problem with the diagnosis is really the detection, or the problem with the detection is the diagnosis. And it is so difficult, and it needs really the expertise of people, that the people don't believe that it's so difficult. It needs really training. But we have something which can help in the meantime. We have the immunofluorescence tests, which make the detection earlier. I didn't show this here. Maybe I have another opportunity in the future. And we can also use the molecular methods, which they can be applied as a complementary methods. We can have PCR, real-time PCR, LAMP, the loop isothermal assay, which I didn't have the time to present here, which has the possibility to detect really nanograms and is very effective. WHO recommended my methodology about LAMP, which has been developed for Cryptosporidium to apply for coronavirus. So there are plenty of methodologies, techniques, which can be complementary here to the microscopy. Microscopy, I would like to say it's very good, useful for the diagnosis, to teach the people from agriculture, from medical schools, from vet schools. And I recommend that all people from any science, they need to learn about emerging disease, about this kind of disease. At least we can teach maybe some modules, few lectures in any part of the faculties in the university, even lawyers, even other people, business people, not only medicals and the related sciences. But now come back to your question. We can use complementary methods, which are molecular methods, but they need evaluation. This is the problem. When you start, this was the experience what I had. Let's say the good luck in my life, it was very stressful, but I went to different laboratories. When I work in Germany, we established in the diagnosis in the laboratory. Many laboratories come to us in Germany. They learned the technique there, and it works when you are in cooperation with the people and you have discussion, conversation. Go to another country, you are using the same things, and you see nothing is working because the quality of the reagents is different. And I had these experiences. So then it's everywhere when you are going to establish the diagnosis for Cryptosporidium, because it's so difficult, you need to cook the situation new. It is like cooking new in a new kitchen. Everyone is making the same food, but the taste is different. And the recommendations now here that the methodologies, they need evaluation. And this is something which is also compatible to the program of the we're speaking today of COST, because the evaluation of the methodology, the technology, they need to be upgraded, they need to be updated. We need to include the new possible techniques which are complementary to the microscopy, in order to improve the technique and develop a simple system that we have cost-effective system. Still very expensive, the diagnosis of Cryptosporidium, and most water companies, they don't like to pay. I know that medical doctors, they can prescribe, and it is about 25 euro to make the immunofluorescence test, but for the water companies and the food material, they need to pay several hundreds of euros. This is not sustainable. And I said also before, there are so many details, I cannot say that we can do this cost effective and in small volumes, but it is necessary to develop, to update the methodologies. And the two protocols which I presented, I said, best promoted in that time, but they are in the meantime new diagnostic tests which we need to evaluate. And this is a very good link, I think, very good bridge to the action of the COST.
No, thank you. Some other question? Okay, well, thanks a lot.
I have one question.
Yes, of course. Yeah, please, of course. Yes, absolutely.
How many people are working in these groups, and are there some people who are working on the parasites, Giardia, Krypton, something like this?
Not of my knowledge, we don't have a lot of people working with the human parasite, but we have a lot of members working with fish parasite, where we are mainly specialized on Saprolegnia, and this, I would say, more historical parasite. We do not have a medical doctor or anatomist, but as I said, this was very interesting and new. And I think also the point that you made about the methods, of course, this is also what we said as a also argument, where somebody is interested and want to join even working on different parasites, because the methods can be interesting, and sharing about experience, of course, and as you say, a bit a sort of a way of making things also working from different places, of course, can be definitely extremely interesting. So thank you. Well, we have a few people that has to leave, so they are also saying, well, thank you, it was a very interesting topics.
Thank you, Serena. Hope to meet you again.
Thank you, Panagiotis. Thank you, of course. It was a real pleasure. Thanks a lot also for your very nice availability and kindness in talking to us this evening. Thank you.
Thank you, and bye-bye.
Bye-bye.